Hey Cyril, you're the only one here, so um, guess I'll get started. So yeah, I'm Sam. I'm excited to present here today my research, phylogenetic analysis of SARS-CoV-2 within coronavirus diversity. And this was completed alongside my co-authors, Ting Chen, Tina, Iris, Bryce, McKenna, and Dr. Baker here at Baylor University last semester. And so as we're all aware of, COVID-19 has emerged as a global health pandemic. And so developing an understanding of how the virus that causes COVID-19, SARS-CoV-2, infects humans is paramount in developing treatments for the disease. And so we analyzed SARS-CoV-2 in three ways. First, an intergenetic way, where we looked at SARS-CoV-2 relation to other members of the coronavirus family. And then second, an intragenetic way, where we looked at variations within the virus. And then finally, we looked at the virus's protein's relation to the human proteome. So that was our network analysis. In order to conduct this research, we obtained a bunch of sequences from NCBI. So we obtained 47 sequences of the coronavirus family, Coronaviridae family, that was for our intergenetic approach. And we also obtained 4 spike protein gene sequences from NCBI. And so the spike protein is basically what's causing the infection. So we're trying to see how does that work across the different coronavirus species. And then we obtained 447 sequences for the intragenetic analysis. So those were all SARS-CoV-2 isolates. And then finally, we obtained 38 proteins associated with SARS-CoV-2 from NCBI for our protein analysis. So in order to do this, I'm going to zoom in a little bit. We used BLASTN. We pretty much used a bunch of BLAST tools. The first one was BLASTN. We ran that on the 47 sequences that were from the coronavirus family in order to ensure that they were less than 85% similar. And the reason that we were doing this is because downstream, when we were doing our tree building and our analysis, we didn't want like 20 different sequences all from the same coronavirus, right? We wanted to have a broad enough diversity that we can say these sequences are less than 85% similar with each other, but these are the ones that are the closest together. So we can see an evolutionary type of pattern. Then we used MegaX to create a neighbor joining tree. And we also created a nucleotide database with the 47 coronavirus sequences and ran tBLASTx using that database and the 4 spike protein genes that we collected. When I say create a database, it was basically like, we have all these sequences, we're putting them in a little file in a folder, and we're gonna let the program do its thing. And then for our intragenetic analysis, we used MegaX to align SARS-CoV-2 complete genome isolates and then develop a phylogenetic tree using UpComa to show the difference between strains. And then for our protein analysis, we used a reciprocal side blast to determine which proteins in the virus proteome most closely matched proteins in the human proteome, or actually the other way around. So which proteins in the human proteome most closely match proteins in the virus proteome. And then after finding the closest human protein hits, we used the string database to develop a network analysis. And we also used a Go enrichment analysis using those same proteins. What a Go enrichment analysis does is all these have associated Go terms with them, which are basically just like they do this thing, like they're transport proteins or they're immunological proteins. And a Go network analysis allows us to kind of figure out what type of those things are most common, right? So which ones are we seeing that we have a lot of transport proteins? Are we seeing we have a lot of immunological proteins from our set of proteins? And so moving on to our results here. So for our intergenetic analysis, we built this neighbor joining tree, and we can see SARS-CoV-2 in yellow, SARS in blue, and SARS in blue and MERS in green. And interestingly, SARS-CoV-2 doesn't seem to be very close to SARS, which is weird considering that it's called SARS-CoV-2, right? But we found that it was actually closest to a bat coronavirus and this coronavirus called human coronavirus 229E. Then for our MSA on our spike protein, our spike genes, we found that the core S protein, the thing that's really doing the infection, is highly conserved across numerous species. So this is pretty much, well, this makes sense, right? Because if it works, if it infects people, like, might as well stick with it, right? And then for our intragenetic analysis, we found that there were three main strains that we identified. So there's this blue one, this pink one, and this green one, using our Upcomud tree analysis. And then finally, for our protein analysis for all of these top figures up here, in this table, we see the results of the reciprocal side blast that got us our proteins. And we can see that there were many viral proteins that mapped to one or two proteins. And I think we identified around 26 different viral proteins. We then used those viral proteins and threw them in the string database. And we got this network interaction. We found that only two of the proteins were really connected with each other. Everything else really functioned independently. And this kind of led us to the conclusion that these proteins appear to work and perhaps even evolve in isolation. For our Go enrichment test, we found that processes related to immunological pathways, they're highlighted. We found that these were kind of overrepresented more than we would expect. Then for our Go term annotation, we found that there's pretty much divided into two main categories. First, transport subnetwork, and then metabolic subnetwork. As you can see, the genes here are in this kind of orangish color, and the terms that they're associated with are in this kind of turquoise color. And so down here, we can see those genes that function more on a metabolic subnetwork. And up here, we can see more of the transport type genes. So why do we really care is really the question. So for intragenetic analysis, we found that SARS-CoV-2, using this analysis, is most closely related to a beta coronavirus. And this really allows us to look at these beta coronaviruses, especially human coronavirus 229E, and see if we can use those same treatments and same methodologies that we used with that coronavirus for our coronavirus that we're currently dealing with. For intragenetic analysis, we found that there are three main strains of the virus. And the largest of these three main strains contain 186 sequences, which was almost half of maybe like a third of our total number of sequences. Oh, I guess, yeah. And keep in mind that these sequences were from all over the world, and they were also done in April. So these results may not actually be still valid, but by using this kind of structure and figuring out where are these three main strains originating, we can determine who's getting the virus, where is the virus located, and which strain of the virus is kind of doing the most damage in a sense. And our human virus analysis showed that these proteins don't really interact with each other, and they kind of led us to the conclusion that they function independently of each other, or they might function independently of each other. And our Go enrichment analysis showed that an overrepresentation of immunological processes may aggravate disease processes associated with cytokine storms. So like when you hear in the news, people talking about how coronavirus causes your immune system to like act up, and the cytokine storm, which often does more damage than the virus itself, our results seem to indicate that fact as well, where you're getting that cytokine storm with these proteins that are similar to the virus proteins. If you have any questions, I would be happy to take them. I know I ran through that really fast, but yeah.
You're good. I did have one question, and you may have already mentioned it, but viruses are ever mutating, right? And they're always changing. So how did you account for that in the study?
Yeah, so that's a really good question. And so again, the caveat for all of this is that our results may no longer be valid because it's been like 4 or 5 months since we did this analysis. But what we did try to do is we tried to list the dates of when we obtained the viruses, and we obtained them all from NCBI, so across the world, people will upload virus sequences to NCBI. And so it's kind of like I wouldn't say canonical, but it has a Actually, well, it can be canonical. So we use the RefSeq database, which is a canonical version of the sequences. So they're curated. People go and like find which sequences are the most common, I guess. That's probably not the right word. But yeah, to answer your question, we pretty much use a public repository and listed the dates when we did our analysis.
That makes sense. So would you have to like redo the study again with more recent data if you wanted more accurate results for like as of now?
Yep, exactly.
Okay.
And then the great thing about like doing this in like computer science type setting instead of using like the BLAST website is that we have our code already available and it's implemented. So you just have to download the thing to a folder and hit go, and it will like do the whole thing again, which is really nice.
Yeah, thank you, Sam. That was really interesting.
Yeah. I think I gotta give you your QR code, right?
Yep. And I turned off the waiting room as well, because I guess that was supposed to be off for these. But it looks like nobody else has joined anyways. I have a question about this that I've been waiting to ask. So with all the different proteins that you show, that you said that they function independently, do you know or did you study what some of those functions are?
Yeah, so we did. I have it. We wrote it in a paper, right? And so let me pull it up because off the top of my head I don't remember.
Gotcha. Also, you said that the spike protein is super similar across all of them, and I mean I've read that particularly between SARS-1 and 2, the receptor binding domain differs pretty, not greatly, but definitely significantly.
Right.
Has that been reflected with your research as well?
So again, like the way that we did our analysis is we took the top 19 most common proteins that were the closest to SARS-CoV-2, right? And as you saw from our intragenetic analysis, SARS-CoV-1 is actually really far apart from SARS-CoV-2, which I don't know, it doesn't make any sense to me why they call it SARS-CoV-2, but
right.
So it makes sense in that sense where the ones that we took to do our spike protein analysis were highly conserved.
Okay, that makes sense. Definitely.
Cool. Yeah, I'm trying to find our, okay. Maybe I'll just like send you this paragraph. It's not really well written, but.
Oh, well, that works. And it looks like people are now starting to join for the next session.